Hi, everyone. Welcome to PLATO. Tom is in Alabama with his family. He's having a great time. Don't worry about him. He'll be back next week, I promise. Tonight, we will have dunes, dust, drought, and downpours, evolution of Great Plains landscapes in changing past and future climates. That'll be tonight at Wednesday night at the lab. Come on over and you can get a second lecture today. And then tomorrow, next week, we have Becoming One in a Million with All of Us. So Samantha Bath from the Institute of Clinical and Translational Research will be here. So I'm going to pass it off to Andy Milkowski.
Thank you. And it's a pleasure to be here. I gave this talk a number of years ago. I think, well, Tom said it, we would have a different audience, but that's not exactly true because my friend Dave Lukey here was present at that one. And it probably was a little bit of a different content. So just a little about myself. I started graduate school here at UW 50 years ago this past fall, and I got my PhD in biochemistry. And then went across town to work in the R&D department at Oscar Mayer. And you'd wonder why a biochemist would wind up working for a sausage maker, bacon maker, ham maker, and type thing. Well, in that era, Oscar Mayer was harvesting animals and they had all these byproducts. And they were generating some pharmaceuticals out of byproducts, and they had a little group called the Biological Products Group, which I was hired in to replace someone who left. But, and this is a career story that I'm sure many of you have had, and that a couple of years into that, they quit slaughtering animals. And my reason for being there was it's going to disappear unless I adapted and they wanted to keep me. Well, they wanted to keep me, so I moved more into the food science research type of area, and in a lot of ways touched all of their products across my career in one way or another, largely in doing a more basic type of work and understanding why things were happening the way they did, which was kind of nice. I called it bucket biochemistry because you do a lot of protein and fat manipulations when you're making products, which is exactly what I studied in grad school. I retired in 2006, and since then, I've been an adjunct faculty member in animal sciences, which is now animal and dairy sciences. And I lecture in some classes. I run the fall semester grad student seminar program and get involved in some extension activities relating to meat processing through our extension specialist, Jeff Sindelar. One of the things I was put into when I was at Oscar Mayer, and I joined them in 1977 in January, was related to what has been controversial since about that time and continuing today. And that's about an ingredient used in curing meat, sodium nitrite. And so I'm going to talk today about that. And a tremendous number of things have been learned over those, 4+ decades regarding those compounds with respect to their safety and in a very, very broad sense. So that's what we're going to cover today. A little bit of sausage making, but a lot more around physiology and medicine and to some degree, people's attitudes and social attitudes. So let's get going here. And for some reason, oops, there we go. Okay, so we're going to start and just do a quick review of curing meat. For those of you who have a chemical background of some sort, nitrite and nitrate belong to a class of compounds called nitrogen oxides, and there are a lot more. Different numbers of nitrogen atoms attached to different numbers of oxygen atoms, and in many degrees, they're interchangeable depending on the environment they're in. So air pollution, it deals with a lot of nitrogen oxides of different sorts. Some of those, one of those, nitric oxide, is one of those compounds. In curing meat, we're taking nitrate, which has the anion form is NO3, negative charge, and we're going to add it to meat, or we used to do that in the olden days. And in the meat, you have a lot of enzymatic activity, which will reduce it, change the charge, take away an electron, so it goes to NO2 minus. So you have 2 oxygens instead of 3 oxygens. Nitrate is very stable, but it can react under the right conditions. Nitrite is much less stable, and it tends to degrade pretty quickly. Well, the second step in meat is that other enzymes in there convert that to nitric oxide, which is a gas. And it has a biological half-life of a few seconds, so it's extremely reactive. But in the case, our case is, it goes and finds an iron atom in a heme protein. This would be hemoglobin in blood, and there's some blood left in the meat that we're using, or a primary protein in muscle tissue called myoglobin. And it sticks to the iron and it changes the color, and it goes, stays pink after cooking. If you were to just cook meat without nitrite or that attachment to the iron, that iron would change this porphyrin ring structure, which is right here. This sits inside of a protein, an iron in the middle. There's an attachment to one of the protein chains, and I'm looking for it. Can't see it here in this depiction, but this changes charge and it changes what's attached to it. Nitric oxide, when attached, keeps it in a raw meat form as a dark red, kind of like raw meat, but in a cooked form, it's pink, as opposed to brown, which is color you see in normal cooked meat. Well, why is that the case? Well, this goes back a few centuries about meat processing. In a few centuries ago, people started processing meat, and they were looking to preserve it over long times, and they learned that if they made this concoction of ground meat and salt and some spices and stored it away, and they ate it later, and a number of people died. It was brown. But if they used a material called saltpeter, which was nitrate, it turned pink and it was safe to eat. The word botulism is derived from an ancient word for sausage, actually. So the organism that caused those illnesses and deaths was Clostridium botulinum. And nitrite or into meat, and nitrite's pretty much the active species forming nitric oxide, inhibits the outgrowth of the C. botulinum organism in the absence of oxygen. And so you don't get that very potent toxin forming. What we've learned in, say, the last half century is that it also participates very greatly in making our meat products safer against another Clostridium organism called Clostridium perfringens, which is related to botulism, but its toxin makes you very, very sick, but doesn't kill you. And that is a risk from eating meats or other foods that are heated for serving and allowed to get to an intermediate temperature and held there too long, and those spores grow out. You eat them, they inactivate in your stomach acid, and they release a toxin at that point, and they make you sick. So a lot of nursing homes where they have these foodborne illness outbreaks are because they're not keeping their warming trays hot enough. They have to be kept above 140 degrees Fahrenheit to prevent that from occurring, because this organism likes to grow between 80 and 130 degrees Fahrenheit. And then the last one is something you probably have heard in the last 20 years is Listeria monocytogenes. And there was a large outbreak of listeriosis associated with some sliced luncheon meats from a plant in Michigan in Christmas time. I forget if it was 98 or 99. And there had been some earlier ones, but they hadn't really registered with people's knowledge. But that organism can grow slowly in refrigerated meat products and dairy products. And it has, if it gets to high numbers and you have the right type of biological situation. So if you're a fetus, that's one of those, or if you're an old person, like most of us in the room, you're more susceptible to getting infected with listeriosis. And if you have a clinical infection, you've got a 20% chance of not surviving. And that includes miscarriages in that. And nitrite slows that growth down tremendously and makes some of the other things that are used to manage that risk in terms of ingredients in processed meats much more effective. And that's a different talk. I could talk about that too. The one thing I want to imprint on you, because we're going to get back to this later, is the amount of residual nitrite in a hot dog, let's say you eat, or 2 ounces of a sliced luncheon meat, or a piece of ham or something like that, is about 0.5 milligram. So when you eat that, you would be ingesting about 0.5 milligram of nitrite. So what we have now in the marketplace is a whole new class of products that are called uncured. They say no nitrite or nitrate added. You've all seen them, I'm sure. If you read the fine print under that no nitrite or nitrate added, which is this middle one, it's in parentheses, except that naturally occurring from blah, blah, blah. Well, companies have, because nitrite in the controversy that's gone on is sort of a dirty word on an ingredient line. Companies have gone and taken dried celery juice, which has a lot of nitrate, and the ingredient suppliers have done some enzymatic or a microbial transformation of that into nitrite, and that's used as the ingredient. And so you have this type of a label. And it's deceptive, but it's also deceptive by requirement of the USDA, because they have standards of identity which say if you don't use nitrate or nitrite in making certain products that are presumed to have those, such as ham, bacon, hot dogs, and some other meats, you must declare on the ingredient or on the front label under or in front of the product name that it's uncured. Behind the scenes, everything's the same, and then you must say no nitrate added. And when this started to come out in the early 2000s, few companies had pangs of conscience bringing this into place, because the competitors who were using it were killing them in the marketplace. So they started doing it, and Oscar Mayer was one of them, but they put in the qualifier there just so they could sleep a little better at night. And actually, this has been over 15-year discussion between a trade group now called the North American Meat Institute and the USDA, saying you have to clean this up because the underlying science is actually back here, and that's nitrite or nitric oxide attached to an iron is what defines a cured meat, and it's the same thing in both cases. But people are misled when they see this. They don't think there's been any of that added. Well, it's still in limbo from a regulatory basis. And there are a lot of competing interests in there. The ingredient suppliers who make these ingredients certainly don't want to see that go away. And, you know, it is what it is. So that's the whole meat science. And now we're going to go into some of the controversies. Why did nitrite get to be controversial and nitrate? Well, ancient times, which was the botulism story, which I just mentioned. There's also a condition in newborn infants who do not have stomach acid developing yet. And secondarily, they have a different form of hemoglobin circulating in their bloodstream called fetal hemoglobin. And that was designed to pull oxygen from their mother via the placenta and has a stronger binding of oxygen than you would say adult form of hemoglobin. But it also has a super strong affinity for nitric oxide. And what was happening was that these infants, and this is about from birth up to about 6 months of age, they were finding out that infants who were eating or at a place where there is high nitrate in the water supply, like around central Illinois, Decatur, Illinois has surface aquifers. Infants were getting cyanosis, meaning they had this, they weren't getting starved for oxygen because the binding of that nitric oxide, which went through that same cascade that I mentioned, because of bacteria in their stomach, no acid to keep it essentially sterile, converted any ingested nitrate into nitrite, went into the bloodstream, got converted to nitric oxide, tied up all of the oxygen carrying ability of the hemoglobin in their bloodstream, and it was cyanosis, same thing as having cyanide, which also attaches to the iron, and that's why it's such a potent poison. And it was called blue baby syndrome. So today, you don't feed newborn infants high nitrate vegetables like spinach and other such materials. And you get bottled water that's nitrate free just to protect them. Well, that occurred in the 40s and 50s. So there was this predisposition or uneasiness about the safety of nitrate in your diet. So that was one step. And then the next thing that happened was the discovery in the 1960s of the formation of nitrosamines. Nitrosamines are known to be in tobacco smoke, but there was some fish meal being treated with nitrate, and it was being fed to animals, and the animals are getting a lot of tumors. And more investigation turned out, and it was these compound, a class of them called nitrosamines, which are carcinogenic. And that touched off a whole look at where human exposure to nitrosamines was occurring in tobacco smoke, in some foods in a variety of ways, in beer, because malting was done with direct air combustion, flame heat, and that was you're having nitrogen oxides coming off of the burners as a byproduct of combustion. It was reacting with some of the components, secondary amines in the malt or in the barley. It was there, and you're getting some small quantities of nitrosamines. Tiny quantities, parts per billion, sometimes parts per million. It was found in some processed meats, namely bacon was a big one. which is fried at high temperatures. And it was a big problem. There was, and this is right when I joined the meat industry, so to speak. I joined Oscar Mayer in 1977, and I was working in byproducts, but about 3/4 of the R&D department was going after this problem. Do we have nitrosamines in our product? Are we going to have to eliminate using nitrite or not? Are there alternatives to keep that pink color and deal with CBOT risk? Are there all kinds of things going on? And a lot was learned, and the regulations were changed about that time. The only product that was really deemed at risk of generating nitrosamines for humans when they were eating it was bacon. And the set of regulations that exist today for amount of nitrate and nitrite you can use in curing meats has a special provision for bacon where it's lower, requires the use of another compound, actually vitamin C, which is a strong inhibitor of nitrosamine formation. And for 30 years, there was a nitrosamine monitoring program in fried bacon with a particular protocol. And that's now being dropped officially from the regulations because by the mid-80s, the entire meat industry had learned how to produce bacon that didn't have this risk. Now, there were a few nitrosamines that were in bacon, but they were extremely low and they weren't deemed harmful. And there are lots of reports here. So there were these National Academy reports, there's pictures of them there. And the final rules were, there were two sets, one published in 78 and one in 86. And these are the rules we have. So if we want to use cure meat, it's broken into different classes, and we're talking about milligrams per kilogram or parts per million if you calculate it. If you're on the English system, it's something like 1/4 of an ounce per 100 pounds, tiny, tiny amounts. But if you're dunking a piece of meat into a curing liquid, a brine with a lot of salt and nitrate or nitrite, you have one set of regulations. That's not done too much because it's an extremely slow way of producing products. If you are injecting with a bank of needles that are penetrating the meat, like ham or pastrami or bacon, and squirting in a liquid containing salt and nitrite, ascorbate, which is vitamin C, and sometimes some phosphates, you are limited to 200 parts per million on nitrite or 700 on nitrate. If you're making a sausage which has ground meat that's comminuted, the numbers are lower for nitrite. For some crazy reason, they're higher on nitrate. I think it relates to fermented sausages in particular, because a lot of fermented sausages are still made with nitrate only. And if you're doing dry cured, where you're rubbing the outside of the meat, you have higher levels, and that's a niche market. Most of the meat made in the US is going to have either upper limit of 200 parts per million and nitrites being used, or 156 where nitrite is being used, except for bacon, which has lower levels where you have 120 parts per million and a mandated 550 parts per million of sodium ascorbate, which is vitamin C, or it's stereoisomer, which is called erythorbate. Low vitamin C activity, but the same chemistry of it. Okay, this thing isn't working, so I got to use the buttons. So then we go to jump forward another decade. And in 1994, the LA Times ran a story in May about a researcher at University of San Francisco. And this was a developing discipline at the time called nutritional epidemiology. Now, some sort of population epidemiology has been done for centuries, where people try to identify, mysterious sources of maladies in humans. All the investigations into COVID are an example. A lot of the public health investigations into food poisonings are other examples. And those are more acute situations, but from a chronic standpoint, diet and health. And that's been going on for a long time. And there's some interesting things you can find in the literature if you go back to the 1800s on diet and cancer ideas. But this researcher, her name was Susan Preston Martin, was the lead author. She was studying brain cancer in children. She was starting to get worried about microwave energy from cell phones being held up to your head. And along the way was looking at diet, and in asking these dietary questions and then analyzing data, was suggesting that there was an association, meaning a statistically significant correlation between mothers when they were pregnant and their offspring when they were around 7 or 8 years old. Mothers were eating cured meats like hot dogs, and their children at that age were having a slightly higher frequency on a population basis of brain cancers. And so that set off the storm, and the whole controversy around the safety of nitrite for curing started up again. And by this time, I was well along in my career in Oscar Mayer, and I'd been drawn into doing the earlier research, and we were looking for substitutes, trying to synthesize substitutes for nitrite. So here we went again, and that brought on a whole another set of scrutiny. One of the things we did, and this is a scientific thing you do, you have some questions. Well, you get a bunch of people together and have a conference and talk it out, talk about what the perspectives are. And so we had a meeting in Georgetown with a number of nutritional epidemiologists. Dr. Preston Martin was invited, but she chose not to come. We had some other people who were doing similar types of work come, and we kind of, from the food producer standpoint, we were learning about what all this nutritional epidemiology was. And so we had a good discussion at that meeting. We also had people from FDA and USDA. And what we learned at that meeting was that as a legacy from what went on in the early 80s, the National Toxicology Program, which is a multi-agency program in the government that does 2-year bioassays, feeding or other types of exposure in animals to look at agents as potential carcinogens, had been doing a follow-up examination of nitrite, and they were in the stage of analyzing data. And then a year later, we had California, which has this Proposition 65 rule. Do any of you know what it is? Okay. California has this unique law where they maintain a list of agents that the state of California and their regulatory agency deemed to be potentially harmful to you in terms of being carcinogenic or a reproductive or developmental toxicant. And that law requires notification of consumers. It doesn't ban the use. It says you have to do that. So you will see on a number of labels, the state of California has determined that blah, 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 may be carcinogenic to you. If you walk into Disneyland, there's a sign right by the entrance because it covers that. Their enforcement of the law is not done by the state, but it's done by the public. And somebody who thinks there is a violation of that can bring it to the attention of their state EPA. If they choose not to start an enforcement, they can sue the cause of that, and they share in the penalties under the law, which are $100. I think it's $100 a day per instance. So if you apply that to a food, it's $100 per day that the package is in the marketplace per package. A huge thing. And they were proposing to list nitrite as a developmental and reproductive toxicant. So I was in charge of an industry committee that we were commenting back to the California agency on that once that started up. Well, in 2000, two things happened. We had our review meetings on the National Toxicology Program and their report and all, and we commented on that. And they gave nitrite a equivalent classification, which means maybe yes, maybe no, due to some tumors in the forestomachs of female mice, and all the other tissue studies had nothing. And these were huge levels of nitrite fed to rats and mice, both genders, over their 2-year lifespans. They were getting up to 2,000 parts per million in their drinking water that entire time. And in Proposition 65, their peer review committee decided that the evidence was not there to require listing. So that was fine. Then the next thing happened with a branch of the World Health Organization in 2006. This is called the International Agency for Research on Cancer, which held a literature review meeting, and they do all of these in Lyon. And they determined that nitrite was a probable carcinogen in their classification system. So more people are worrying about it. 2007, there's a charity called the World Cancer Research Fund. They put out reports. They're closely linked with some regulatory agencies around the world. They put out their 2007 report. They do a full one every 10 years and updates in between. And they were saying, we think any cured processed meat is risk for colorectal cancer, so you should worry about it. Meanwhile, the IARC determination had to do with stomach cancer, different cancers. And then in 2014, California came back on their carcinogen side and was proposing listing, and they haven't listed it for a variety of reasons. Some legal, in terms of who has jurisdiction on what's on a label, USDA, which is a federal level, or the state. The state doesn't own the label within on anything in interstate commerce for meat products. Food products is different, other food products. Okay, so I bored you to death about that. This is the interesting part. The other stuff about nitrite. This was not known at all when the controversy started in 1977, 79, 80, early 80s. And it's a fascinating story of what you learn on with time going on. First of all, you secrete nitrate in your saliva. Wow. Who would have thought? Well, your salivary glands are going to suck nitrate that's in your plasma circulating around, and they're going to deposit it into your mouth. And that's going to be reduced by a biotransformation from the bacteria in your mouth, all those commensal bacteria. And you're going to make nitrite and you're going to swallow it. Interesting. And if you do sort of the input-output balance, probably 90+% of the nitrite that you swallow is coming from that recirculation and your water. And somewhere around 5 to 8% is coming from nitrite and cured meats. And so you'd say, well, why in the world would you evolve to have that going on in your body? Well, the answer, and it's not a absolutely known thing, but it's the strong theories around it, is that nitrite has a biological benefit. First of all, it protects you from some periodontal disease. And then second, when you swallow that nitrite and it hits your stomach acid, it's going to break into some nitric oxide and some other nitrogen oxides. And those form nitrogen oxides, mainly the acidified nitrite and the nitric oxide, are strong enhancers of the ability of the low pH acid in your stomach to kill those harmful bacteria that you're also bringing in with all your food, especially the raw commodities. Interesting. That's been proposed to be a form of innate immunity, that we evolved to do that as a self-protective mechanism. And there's a number of papers on that. And this is the balance. And I actually calculated this, and we published it in a chapter in a book a little over 10 years ago. So if you take the diet, except for any cured meats, you have a huge range of nitrate exposure. Where's it coming from? Lettuce, radishes, potatoes, beets, red beets, celery. Let's go back to the label, right? That I was showing you earlier. There's a little bit of nitrate from some cured meats. It's going progressively down because you're seeing more just nitrite used. Highly variable amount from water, depending on where you live. If you're getting your drinking water from surface sources and you have agricultural fertilizer runoff, you'll have higher nitrate. If you're getting it from deep wells, you'll get a little bit, but not so much. So that's why it's got that big range. And then because of the recycling and depending on how many vegetables you're eating and what your water sources is, you can have a huge range there. And then the last line, endogenous synthesis. When I was in grad school, there was this mysterious substance that was called the endothelial relaxation factor. It allowed smooth muscle, which is controlling your blood pressure, to relax, and nobody could figure out what it was. Turns out it's nitric oxide. And the body synthesizes nitrate and nitrite, which I'll talk to you in about a second. But back to celery as a source of your exposure to nitrite and comparing that to cured meat. I was on an airplane trip going somewhere, and they passed out a snack. I got a little packet of celery. And I had been working on this for a while, and I said, high nitrate. I wonder what's going to go on with that enterosalivary recirculation route. So I looked up the amount of nitrate in celery, which is around 3,000 ppm. It's highly variable depending on type of celery and fertilizer use and all of that. But 3,000 parts per million is a lot. That's 10 times higher or 20 times higher than you use in curing meat, but it's all nitrate. And so I said, okay, well, what if I eat all the celery in a 45-gram snack pack? And I said, okay, that 3,000 ppm, it's going to have 0.135 grams of nitrate. I'm going to swallow it. and it's going to recirculate and come back into my saliva as nitrate, about 20% of it. And about 20% of that's going to convert to nitrite, and I'm going to swallow that. And that nitrate that's still there is going to go back into my stomach and then recirculate again. And I just did 2 cycles. And so the net on that is I'm going to be swallowing 6.5 milligrams of nitrite from eating that 45-gram pack of celery. And if I eat about an equivalent amount of hot dogs or sliced ham, I'm going to eat somewhere between 0.5 and 1 milligram of nitrite. So is it harmful or not in the grand scheme of things when the bulk of my dietary or ingestion exposure is going to come from something other than a cured meat? Good question, huh? Well, what was discovered in the mid-'80s was that we synthesized nitric oxide. It breaks down very quickly to nitrite, and then that breaks down to nitrate and gets excreted or recirculated in the body some way. Nitric oxide and nitrite have very, very important physiological functions. They're profound. Nitric oxide is a neurotransmitter. It's involved in the smooth muscle relaxation. It's involved in immune response. So your macrophages will generate a ton of nitric oxide and release it at a spot where they see there is a foreign object coming in that you got to fight against in order to kill that cell, inactivate it with the blast of nitric oxide. And this was all being learned in the mid-'80s on. Fascinating. You know, in our lifetimes, this was totally unknown. So, and in fact, Science Magazine, which is sort of like the Time magazine of the science world, called it the molecule of the year in 1992. And they had this headline, "No news is good news," which is sort of a play on words. And then we've got drugs that are used for various purposes, and they release nitric oxide into certain tissues. The oldest ethical pharmaceutical known as nitroglycerin, used to treat angina because it relaxes smooth muscle tissue, heart muscle tissue. And that's from the 1860s. And more recently, Viagra. So who would have thought? There was a Nobel Prize awarded in 1998 to a group of researchers working on nitric oxide physiology, and you can look up that and for their pathfinding work. And we now talk about a nitrogen oxide cycle, where we've got the internal synthesis, which is in here. NOS stands for nitric oxide synthase, and it takes arginine and goes through a whole metabolic pathway and interconverts with nitrite, winds up in the blood, goes to nitrate, comes into the oral cavity, goes to the stomach, and all kinds of good stuff. It's amazing. But we're afraid of nitrite in cured meats. Okay. When I gave this seminar to the grad students in our department last fall, and so I had done this many, many years ago. I updated this and I went to a website called clinicaltrials.gov. It's maintained by NIH, and it's a database of all the clinical trials that get listed anywhere for whatever purpose. And I type in the word nitrate, and when I first did it in 2016, there were 250. Well, 6 years later, there's 700. And you look at nitrite, 700 on nitrite. last fall and 2,000 on nitric oxide. There's a tremendous amount of medical work going on to use this to advantage. And there's some interesting ones there in terms of deliberately giving people my age, I'm 72, sodium nitrite pills to take about 20 milligrams a day to relax smooth muscles and keep your blood pressure down and all that good stuff. And that one, I'm not sure if it's gone anywhere. When I looked at it, they're just saying no adverse effects. I mean, so far. So that's what you've got. So I'm going to wrap this up. And then, but before I do that, we had this up for a purpose. We talk about exposure to nitrite. Anybody use this type of sensitive teeth toothpaste? Okay. Did you know that this has 5% potassium nitrate in it? That's 50,000 parts per million. Okay, and yeah, you take a little dab and you don't swallow it, at least not much of it, but this probably what you swallow is similar to that celery example, or on that order every day. I brought this from home. It's an unopened package, but you can go look at it, what's on there. We don't even think about that. And then when I'm officially done, and we'll probably stop the recording, I've got a little exercise for testing the nitrite and nitrate in your own saliva here. I brought some testing strips here, and we'll go through that. So these are just few general conclusions, a little bit of certainly my opinions from throughout all of this. And there are some people who, are still very, very worried about nitrite as a toxicant. But we know from the last 30, almost 40 years of nitric oxide research and what's been discovered and what continues to get discovered, that a nitrogen oxide metabolism, nitric oxide, nitrite, and nitrate are essential for our own well-being, our memory, our blood pressure, our ability to fight off infections, okay? And the nitrate that we ingest and the nitrite that we make in our own metabolism or in situ somewhere in our bodies, in the stomach or in the lower intestine, and that those could be forms of innate immunity. It's not a well accepted or universally accepted hypothesis, but I think it's pretty well thought that, yeah, it's probably true. It's not well known, I would say. Those low levels of nitrate and nitrite we use in meat curing are safe. And what they do for food safety, I think, makes it appropriate to use. The questions we had in the late 70s through the 80s, they were very appropriate. We didn't know what was going on, but we've learned so much about this whole area in that time that I think we can feel pretty comfortable about it, okay? And you can never say you have no risk, that doesn't exist, but I think the risks are truly very low to us from using that or from having cured meats. So, and so I would just say, eat your cured meats and don't worry about it. We're okay. So, but we've got the other point of view, and you have to respect that. So in general, consumers are concerned about additive. Anything that's synthetic, has a chemical name, people are worried about these days. I grew up in the era of better living through chemistry. Well, it's the other side of the pendulum right now. And it's interesting that in the US, we don't take the time in our news media to kind of go down and dig and try to explain things on a more technical basis. It's really, really hard to do, but it can be done. If you go listen to the British media about this, they're amazing in that they try to dissect a situation and do the steps along the way, whereas we get the high-level discussion and said, this is bad for you, and you get somebody who says, yeah, it's really bad, and you get some other person saying, no, it's good, and they say, well, they're in industry, so they don't count. And that's the level we discuss it. We don't go down to the more mechanistic technical discussion. It's just not in our culture here in the US for whatever reason. We do have regulatory loopholes. This calling cured but uncured, or I call it alternatively cured meats. Those exist, and they're everywhere in all the rules. Look at our tax code, and people are smart. They're going to take advantage of those types of things. And our regulators are really confused. And I don't say this to be mean, but I say they have such a tough job. And their position is trying to be protective of their clients. And if you're the USDA, you don't want to have anything bad happen to anyone eating meat products or anything else. So they are going to be ultra conservative, and so they're going to take this principle called the precautionary principle. If there's any doubt, we're going to err on the safe side of caution. And Europe is much more strongly in that direction than we are, but that's going to generate the controversy too, because it's going to slow down doing other things if you're trying to deal with adhering to regulations or finding the loopholes or ways to get where you want to go while adhering to the letter of the law. So that's the world we live in. So more research going on. What you see in medical school textbooks today still has a high amount of the 1980s concerns about nitrite and nitrosamine safety, and it's creeping into textbooks now. The nutritional sciences and nutritionists are kind of getting there, but not very quickly. So you're still going to get these messages about nitrite is bad for you. And that leaves us in a situation where the public's really suspicious about nitrate and nitrite. But I'm here to tell you that our exposure to nitrate and nitrite is essential for life. And we're going to get those from all the foods we eat. So don't worry about it. Just go on your merry way. We're doing okay. So thank you very much. And I'm happy to entertain some questions. And then when we turn off the recording, we'll get to looking at nitrite and nitrate in your saliva. So, yes.
I just have a question. Those old studies that show that cured meats, all these health benefits. You think those were?
Can you say that again?
The studies that that said that there's all these health.
Oh yeah, health consequences. Yeah.
Cured meats.
Yep.
You think that was just science, a case of maybe correlation, not causation?
Correlation, not causation. So that's another lecture. You ought to have me back to talk about epidemiology 101. Okay.
They're ranging from 50s to 70s.
Exactly. And just to give you an idea of how difficult it is to do, and there's quite a bit of human nature in this, in that you try to be neutral in setting up a study, but if you've got suspicions, you're a little predisposed to finding something. If you find something, you publish it. If you don't find something, you don't publish it. So the negative results you never hear about. That's one part. So it's a publication bias. You only hear the publications where they've found an association and it's statistically significant. But there is a gulf between people who produce foods and really know what they're doing in terms of how they classify things and do stuff, and the people who do nutritional epidemiology. So I'm going to ask you a question. First part of it. What did you have for lunch on January 17th? Okay. A lot of these studies are done on dietary recall. So you got a questionnaire with, 100, 150 questions at most. And you get questions that are classified across the board, all kinds of foods. And the way they'll talk about meats or cured processed meats, you can question how well they categorize things well, because they're not done by somebody who knows how you classify meats at the level of understanding that I was talking about earlier. But on top of that, they ask the question once or once a year over a number of years as they track a cohort. And there are different ones that they go get a bunch of people who had one type of cancer, and they start asking them about their diets. And then they try to match them with folks who didn't have cancer, ask them about their diets, and try to explain the disease state in terms of a difference in the diets. It's highly imprecise, plus the accuracy of the dietary information is really not super good. They're getting better, but they still have such a long way to go. And they calibrate themselves versus these questionnaires being compared to 24-hour recall, where you remember better, on a subset of them. But if you look at how many of you have been exposed to statistics? Statistics. Okay, you know what a statistical test of significance is? Okay, and you look for 95% confidence. That's one thing. And you look for a value called R squared. And the value of R squared in that is how much of the dependent variable, in this case, cancer, can be explained by the independent variable, which would be, say, eating a cured processed meat, or and they apply it to the accuracy of their dietary recalls. And if you compare a 24-hour recall against these other general recalls, the R squared of those is somewhere between 0.6 and 0.7. No, R squared is somewhere between 0.3 and 0.5, means that they're 50% accurate at best. So flip a coin on those. And you try to challenge nutritional epidemiologists, and they say, oh, no, you don't understand. You're not too accurate on that. The other thing is you have these odds ratios that come out that say the increased odds or risk level of having a malady based on what they determine is significant. And the error around those average values is huge. And in every case that I've seen, as you go along and study that more, that odds ratio gets down to the point it becomes statistically insignificant. 15 years ago, or back when this work came out from maternal consumption and childhood brain cancer, they were talking about relative risks. Odd ratios are slightly different, but essentially the same, of on the order of 1.8, 1.9, which means you have a 80–90% higher risk if you are in this group that's, consuming cured meats versus someone who doesn't. Okay, go to those studies now. The last one's coming out. It's now 1.1. And it's at borderline significance. Studies have gotten larger. They've looked at more people. And it says we really need to be careful. And then the other part is, in the 90s when this started out, nutritional epidemiologists were really saying, if you have a relative risk under 2, all of our methods for determining these things are so imprecise. And there was an attitude, a decision, that that's probably, you can't say anything. It's highly equivocal. Well, today you get a statistical significance of 1.1, and somebody's out trumpeting that that's the cause of the illness. And I'm bothered by that jump because it's still probably very, very equivocal and not there. There certainly could be effect for a subset of a population between ingesting sources of nitrite, cured meat being one of them, and getting cancer. It might be due to genetic, some other lifestyle, total diet, other things. And that relative risk for that tiny, tiny subset might be 3 or 4. But we don't know how to identify that subset. And so over the entire population, you get a 1.1. Okay. So what do? Do you eliminate them and for some unknown subset of the population for that? And then the other counter argument is that, okay, so you have a relative risk of 1.1, but it's small, it's tiny, but take it over the world's population of 5 billion people. That's a lot of people. So we ought to be doing something about it. The problem is you can't tell who you ought to be doing something about it for, which is a social problem, not a scientific problem. Sorry about the long-winded answer. But I can give you there's a set of criteria to judge the quality of nutritional epidemiological studies. And I can bring in some examples. I've given a lecture on that to some students on campus.
Yeah,
Yes, sir.
A little tangential to everything going on here. This is the nitrates in solution in your body or in the food. But how about in the air? Is anybody done any? I was just thinking nitrous oxide. Lactic gas that does something. But just general, say nitric oxide pollution. Is that do things to people?
Yeah. I mean, that's highly involved in if you're breathing in some nitrogen oxides, they will do things to your airways or your bronchi and affect your ability. They'll contract or expand them. And I'm not up on that, but I happen to have asthma. And one of the diagnostics you go in is, how much nitric oxide do you exhale? And so, yeah, it's something to be concerned about, and certain people can be acutely sensitive to that. And chronic exposure, I'm sure it might not be good for you.
Did you discuss nitrosamine? That forms in meat, correct?
No. Oh, yes, and no. Okay. Nitrosamines require high temperatures. So I'll tell you the regulatory protocol for measuring nitrosamines and saying when you're out of compliance. It's on bacon. You take the bacon 21 days after packaging, you fry it on a very tightly controlled, temperature controlled griddle for 3 minutes on each side. And then you send it off to a lab and they do a nitrosamine analysis. If you are over 17 parts per billion total, and then there's individuals of the few of them, you're presumptive, and you have to do something to lower that in your bacon processing. We had a lab where we were measuring nitrosamines until the late 90s and then quit because we're getting everything way under that. That would be considered a harmful level. And if you do feeding studies of nitrosamines or exposure studies through a variety of means, yeah, you can develop a number of tumors. So.
So you were talking about a subset of things that could be more sensitive to nitrates and possibly nitrosamines.
Yeah,
There's no way to determine that, but
we don't know how to do it.
studies that are now being done with large numbers of people with and without cancer and other conditions could potentially identify groups of people that are more sensitive and have specific genetic.
Yeah, there might be, but I'm not aware of any, but that's not my specialty at all. So. Mhm.
You know,
So certainly, yes.
Do the nitrosamine, do they just form on meat?
No, no, you can get them in a number of other foods. It's a reaction, it's temperature dependent. You have to have frying temperatures that, like, the protocol is 340 degrees Fahrenheit. If you're, and that's the other thing, most products, you worry about this, but if you're cooking, if we cook hot dogs, Dave, what were we cooking hot dogs to in the wiener tunnel? Yeah, 160, I think there was 178 coming out below the boiling point of water, and the air temperature was right at about 200. If you're making ham, the air temperature inside of an oven where you're cooking ham is going to be about 180, or if it's in a water tank, it's going to be about the same. You're well below the temperatures. Now, your point about cooking hot dogs on a grill, that's an interesting one, because you look at the thermodynamics of cooking, mass and the surface area and all. And yeah, the grill is very hot, but you are limited to the temperature on the surface of the product to the boiling point of water until you evaporate it in a substantial amount of water, and then it goes up. It's the thermodynamics. So bacon, because it's low moisture and higher fat, gets up to that 340 on a griddle really fast. But a hot dog on a grill or a hamburger on a griddle where you've got a lot of water, and a hamburger doesn't have nitrite to it, you're evaporating so much water in the cooking process that is evaporative cooling, and the surface temperatures are much more moderate. And so you're not getting nitrosamines forming to any great extent in that. And that's why we had no regulations on those other classes of meat products. So